I am Elhanafi Khulud. I am PhD candidate.
My name is Silvia Keipo. I'm postdoctoral researcher at the University of Po.
I am working in analytical techniques for understanding the mechanism of interaction of mercury and selenium in living organisms.
I'm working at the research team of David Ramo and Zoina Pedrero.
Merfish project allows me to work in different samples, especially in some fish samples in collaboration with INRAE. And we can work on the internal tissue of this fish, that's the muscle, the liver, the brain, and other tissues, for understanding the mercury and selenium interaction in this tissue after a 6-month exposure of two different food diets, a tuna-based diet and another alternative vegetarian diet, to see how mercury and selenium will be after exposition.
The main objective of Murphy's project is the evaluation or assessment of the interactions between mercury and selenium in biota. In our case, it's mainly developed for fish samples.
The problem is that in fish, in all the animals in the ocean or in the aquatic system, they can be contaminated by mercury after the cycle of mercury. And this human concept is this fish and this seafood, and we can have this contamination in human also. So understanding the mercury behavior in this living organism, especially after a co-exposure with selenium, will enable us to better understand this interaction and detoxification of mercury in this living organism, and to avoid that human can be affected by this contamination.
Of course, mercury is considered a potent neurotoxin, especially one of the compounds of mercury, methyl mercury, that can generate neurodegenerative problems. Mercury and selenium present a high affinity, a high chemical affinity, and this high affinity causes an antagonistic relationship between toxicity antagonistic relationship between both. So this means that in the organism, selenium can capture, or mercury, well, we don't know exactly which captures the other one, but selenium can capture mercury, reducing its viability and its mobility inside the organism.
The staff exchange, it allows a lot of my colleagues to go and to be abroad for some mobilities, to solve some questions that they have. For example, if we cannot make the experiment in our lab, or we need like an equipment that is in another lab that we have a cooperation with them, we can go. It facilitates this task.
If we consider the partners of the Murphy's project, they are research groups that are in the top of the mercury field research.
I have the pleasure to work with Tamara, that she comes from University of Madrid. She works with me in my sample in the subject that I am working with. With her, we can go faster in the sample preparation, treatment, and analysis. Also, which was able for her to see a new method for the analysis of mercury and selenium, especially that she works also in mercury and selenium, but in other fish species. So to work in aquaculture fish, it was a great task for her, and it expands the collaboration between labs, academic or industry, in Europe, in Brazil also, and in different countries and collaborators.
And at the end, we can make a mixture of all this knowledge and build new knowledge based on the different experiences, because all the people working here are in the top of the mercury world.
It allows you to be more able to adapt to new conditions. When you go to a new lab, industry or research, you'll be in a new team, a new equipment. It's not the normal condition that you go every day to the same lab, and this is very interesting. You will work with the new people, with a new team, and you need to be very able to adapt to the new situation, and it is very important during this mobility and exchange.